Hi, my name is Joanna Ficek and I'm a PhD student in the Biomedical Informatics Lab at ETH Zurich. Today, I'd like to present you our work on single-cell integration via matching, for short SCIM. This work will also be presented by my co-first author Stefan Stark on Tuesday, July 14th. So if you'd like to know more, I encourage you to join his talk. But now I'd like to walk you through our poster. So what's the motivation for our work? Recent technological advances enabled studying samples such as patient tissue at cellular resolution. Typically, a sample consists of different cell types, here represented by different colors. Although it's a heterogeneous cell mix, most cells will be present in several copies. In order to study the sample on various molecular levels, multiple omic technologies can be applied, here represented by shape. However, molecular profiling with each of these technologies often consumes the sample, and the exactly same set of cells cannot be profiled with yet another technology. Therefore, after multi-omic profiling, data from the same sample or cell mix becomes a set of disjoint datasets. These datasets lack both cellular and feature correspondence. This makes the integration challenging since most of the available multi-omic integration methods rely on such correspondence. However, recovering the lost cell-to-cell correspondence is possible, for instance, with SCIM. SCIM is a framework combining deep learning with bipartite matching. It assumes an underlying latent structure that is shared across technologies. This assumption is valid in all kinds of experiments where a sample is uniformly distributed across technologies for analysis. SCIM integrates single-cell data by firstly constructing a technology-invariant latent space, and secondly, matching the cells across technologies using the obtained embeddings. Let's see each step in more detail. In the first step, a latent space is constructed using a set of autoencoders, one per technology, and the technology-specific latent representations are aligned with each other. The loss is a weighted sum of the two components. The negative log likelihood measures the reconstruction error and ensures faithful representation of each technology. The second component is an adversarial term to align the latent representations across technologies. For this, we use a discriminator that classifies the latent embeddings into their original technology. By fooling the discriminator, the latent space becomes aligned. This adversarial autoencoder framework constructs a technology-invariant low-dimensional latent space. Importantly, obtaining a shared latent representation doesn't require any correspondence in the data space. In this well-integrated latent representation, analog cells profiled with different technologies lie in close proximity. Therefore, in the second step, these embeddings, marked with Z on the figure, are used by bipartite matching to find cell analogs across technologies. The data from two technologies, here circles and hexagons, can then be represented as a graph, where each cell is a node. To increase the computational performance, the search space is first reduced to a set of potential analogs by k-nearest neighbor search across the technologies, as indicated by the dashed lines. The Euclidean distance between embeddings is used as the cost on the edges between each pair of cells. To account for the possibility that the cell distribution may not be exactly the same across technologies, the KNN graph is extended with a null node. It captures the cells for which a correspondence could not be reliably recovered and prevents mismatches. We then phrase the problem as min-cost-max flow with the objective of identifying maximal number of cell analogs at the minimal cost. This gives a set of matches represented by fig lines in the figure. To investigate the performance of our framework, we use simulated as well as real-world data. Firstly, we use three simulated single-cell datasets with a common branching structure marked by color code and a letter, as well as a fine-grained pseudotime information which represents progression through some process. Importantly, the simulated features are not shared across the technologies to better mimic real-world multi-omic studies. SCIM creates a shared latent space between all the technologies, which is oriented using the branch label. The shared latent space is visualized here by t-SNE, and each column shows only cells from one technology. Different technologies occupy the same region, which suggests that latent space integration scales well to multiple technologies. In the upper row, points are colored by their discrete branch label, and in the bottom row, the color code corresponds to pseudotime. Not only data clouds corresponding to different branches are aligned between technologies due to supervision, but also the fine-grained pseudotime information is consistent across technology embeddings. In the second step, Cells from each pair of technologies are matched using the bipartite matching scheme operating on these joint embeddings. The density plot shows alignment of the pseudotime information across matched cells between two technologies, source and target. SCIM accurately matches cells with the overall branch label accuracy of 86% and aligns their pseudotime as depicted on the figure, giving Pearson's correlation coefficient of 0.86. We also apply SCIM to a couple of heterogeneous real-world datasets. Here we show a latent representation of a melanoma sample profiled with single-cell RNA-seq and CyTOF technologies, resulting in more than 100,000 cells. Semi-supervision using only 10% of the labels is used to orient the latent space. On the T3 embedding, we can see clusters of B cells in blue and T cells in orange, with the cell matches marked by gray lines. Despite the unbalanced cell distribution, the bipartite matching results in the cell type label transfer accuracy of 93%. In conclusion, we developed a method to restore lost cell correspondences in multi-omic datasets. In the first step, SCIM constructs a shared technology invariant latent space using a set of autoencoders. In the second step, it matches cells across technologies using min-cost-max flow framework. Thanks to the employment of deep learning, SCIM handles lack of feature correspondence and scales to large datasets and multiple technologies. Such computational integration of single-cell datasets allows to take advantage of the complementary information generated by different omic technologies while preserving the cellular resolution. With that, I'd like to thank you for your attention, and if you'd like to find out more about the method, simply scan the QR code. Thank you.